electroencephalogram. The EEG pattern of REM sleep bears striking resemblance to those of wakefulness, and individuals who are awakened from REM sleep often report vivid dream experiences. Subsequently, Dr. William Dement of Stanford University independently identified a similar sleep state characterized by low voltage EEG patterns and ocular motility in cats. Michel Joubert subsequently replicated these findings and additionally discovered that REM sleep is accompanied by a loss of muscle tone, namely REM atonia. Due to this apparent paradox of EEG patterns, EEG patterns resembling wakefulness while the organism remained in the sleep state, this stage was sometimes dubbed as paradoxical sleep. As you know, the stages of human sleep and wakefulness can be differentiated through the use of EEG, electroencephalography. Non-rapid eye movement sleep or non-REM sleep is conventionally divided into 3 distinct stages. It's worth reiterating that REM sleep is characterized by low amplitude EEG, which bears resemblance to the wakefulness. This observation suggests that the cerebral cortex is desynchronized during REM sleep. This diagram illustrates a comparison of brain activity during rapid eye movement sleep and wakefulness as measured by PET or functional MRI. A highly specific pattern of neuronal activation is discerned during REM sleep, characterized by heightened activity in several regions of the limbic system, such as amygdala, hippocampus, and anterior cingulate gyrus. It's noteworthy that the amygdala, in particular, demonstrates increased activity during REM sleep. So, as previously discussed, muscle atonia is a hallmark of REM sleep, allowing our bodies to remain immobile despite the activation of numerous regions in the cortex. The regulation of atonia during REM sleep, or REM atonia, is accomplished through a descending pathway stemming from the brainstem. Specifically, premotor inhibitory cells in the ventral medulla send inhibitory projections to alpha motor neurons, resulting in inhibition of these motor neurons. The mechanism of atonia is impaired in human patients suffering from REM sleep behavior disorder, or RBD. Individuals with RBD exhibit incomplete or absent paralysis during REM sleep, leading to the acting out of dreams through behaviors such as talking, yelling, punching, kicking, sitting, and jumping from bed. This disorder is believed to stem from impairments in the brainstem systems responsible for inducing atonia during REM sleep. In a recent study, we demonstrated that the neuronal pathway comprising glutamatergic neurons in the SLD, the pontine region, specifically the S-subcellulus or SLD region, and glicinergic neuron in the VMM, ventral medial medulla, that send projections to inhibitory projections to anterior horn of spinal cord, in which alpha motor neurons reside, and plays a crucial role in inducing REM atonia. And furthermore, we also discovered that this pathway, SLD, BMM, alpha motor neuron pathway, is also involved in inducing cataplexy in narcoleptic mice. So let me just briefly mention about narcolepsy. Narcolepsy is a neurological disorder characterized by an inability to maintain wakefulness states, most notably daytime hypersomnolence or daytime hypersomnolence, which often leads to sleep attack, which is a sudden onset of non-REM sleep at inappropriate times, such as during conversation or operating machinery. In addition, patients with narcolepsy may also experience episode of cataplexy, which is characterized by a sudden weakening of posture muscle tone triggered by positive emotions like laughing or excitement. Cataplexy is thought to be a pathological onset of REM sleep or REM-related phenomena like REM atonia during wakefulness. In the interest of time, I won't bore you with the detail, but we have found that cataplexy is induced by the same SLD-BMM motor neuron pathway involved in REM atonia, thus indicating that REM abnormality is a characteristic feature of narcolepsy. Please read this paper if you are interested in our findings. And as you may know, human narcolepsy has been shown to be associated with specific loss of hypothalamic orexin neurons, as demonstrated by a study conducted by a research group at Stanford University, Dr. Emmanuel Mignot's group. This study revealed that narcoleptic brains lack orexin peptide. And this is shown by in situ hybridization study. So as a result, narcolepsy is now understood to be a disease caused by the loss of orexin-producing neurons in the hypothalamus. So it is widely accepted that monoaminergic neurons play a pivotal role in regulating wakefulness. So specifically, 5-HT neurons, serotonergic cells, and noradrenergic cells, and histaminergic cells are localized in the brainstem and hypothalamic regions and diffusely innervate the whole brain. And these cells fire at rapid rate during sleep, during wakefulness, and slow down during non-REM sleep, and almost cease firing during sleep. So wakefulness is thought to be maintained by these monoaminergic systems. And orexin neurons are known to send excitatory innervation to these monoaminergic cells. So monoaminergic neurons are typically active during wakefulness. However, the firing patterns of dopamine neurons within the ventral tegmental area diverge from those of other monoaminergic neurons with regard to sleep and wakefulness state. Studies have reported that BTA dopamine neurons exhibit burst firing during REM sleep, while other studies have suggested that these neurons exhibit no discernible change in mean firing rate across the various stages of sleep and wakefulness. More recent fiber photometry data has revealed that BTA dopamine neurons exhibit a lower rate of firing, lower rate of calcium transient during non-REM sleep in comparison to either wakefulness or REM sleep. Furthermore, a recent tracing study conducted using viral vectors has disclosed that the BTA dopamine neurons consist of heterogeneous populations with differential input and output organization, indicating that potential existence of multiple BTA dopamine neuronal populations with distinct roles in sleep wakefulness regulation and defining patterns across sleep and wakefulness states. Thus, we conducted an examination of the extracellular levels of dopamine across the sleep and wakefulness states in various brain regions using fiber photometry with GPR activation-based dopamine sensors, GRAB-DA sensors. The goal of this study was to determine the existence of subpopulations of VTA dopamine neurons with differential roles in sleep and wakefulness regulation. So the research group of Dr. Euron Lee at Peking University developed the GRAB-DA sensors, which allowed for the reliable and specific measurement of dopamine changes with high spatial temporal precision. In response to extracellular dopamine, GRAB-DA sensors exhibit substantial increases in fluorescence with subcellular resolution. In order to perform our examination, we expressed the GRAB-DA sensor in the basolateral amygdala, BLA, nucleus accumbens, and the medial prefrontal cortex and lateral hypothalamic area of mice by injecting adeno-associated viral vectors carrying GRAB-DA into these designated regions and implanted optical fibers in these corresponding regions to monitor fluorescence to examine the relationship between extracellular levels of dopamine in these regions and each transition between sleep and wakefulness states. The level of dopamine in the BLA, basolateral amygdala, exhibited a distinctive pattern and characterized by a transient increase immediately preceding each non-REM to REM transition and a decrease during REM sleep. This is the representative pattern of DA dynamics in the BLA in mice. And these panels depict the average DA levels at each transition between vigilance states, including non-REM to REM, REM to wake, wake to non-REM, and REM to non-REM sleep. These panels are presented as heat maps. As you can see, as can be observed via electroencephalography, non-REM to REM transition occurred at the time when peak DA levels are observed. So in the nucleus accumbens, the level of dopamine also displayed a slight increase, slight elevation immediately preceding the non-REM to REM transition and fluctuations during REM sleep with a slightly elevated mean value during REM sleep. The medial prefrontal cortex, MPFC, and lateral hypothalamus exhibited similar patterns of dopamine characterized by pronounced decrease of dopamine level during REM sleep, without preceding increase of dopamine levels. In contrast to dopamine, the levels of norepinephrine, noradrenaline, and 5-HT, serotonin in the BLA were decreased during REM sleep. And began to decrease during end of non-REM sleep, and began to decrease further shortly prior to transition to REM sleep and remaining at low level during REM sleep. So we discovered that dopamine levels were increased in the BLA immediately preceding non-REM to REM transitions, displaying different patterns in other regions. To investigate the physiological significance of the BDA peaks in the non-REM to REM transitions, we employed an optogenetic approach. We utilized stabilized step function opsin SSFO in BTA-DA neurons in that iris cream mice using a QRI-dependent AB vector. Now, as we found that optogenetic excitation of DA neurons with SSFO by applying a light pulse of 1 second caused the transient DA elevation in the BLA in a similar time course to DA peaks that we observed at the non-REM to REM transitions. And then we bilaterally implanted optical fibers in the BLA. And optogenetic excitation of DA fibers in the BLA during non-REM sleep, caused a transition to REM sleep, which began about 120 seconds after stimulation, which was significantly shorter than the control. Additionally, the duration of REM sleep after the stimulation was increased as compared with control. Excitation of DA fibers in the BLA by applying light pulses every 30 minutes from ZT8 to ZT11 laboratory increased REM sleep amount at the expense of non-REM sleep. The power spectrum of EEG during each state was not affected by the manipulation. The duration of non-REM sleep was shortened as non-REM sleep bouts were terminated earlier than usual. At the same time, we observed an increase in host-positive cells in the BLA after the manipulation, indicating the activation of amygdala after stimulation. In contrast, the stimulation of DA fibers in the nucleus accumbens prefrontal cortex or lateral hypothalamus didn't alter the duration of REM sleep or the latency of the transition from non-REM to REM sleep. We further evaluated the effect of inhibiting the BTA-DA BLA pathway on REM sleep. For this purpose, we employed vertebrate low wavelength opsin, VLWO, which couples to the GI subclass of G proteins, and transiently induces hyperpolarization by applying light. We delivered AV DIO BLWO in BTA-DA neurons and implanted an optic fiber in the BLA of that iris cream mice. After stimulation during non-REM sleep, the latency from non-REM to REM sleep was prolonged as compared with control. And the REM sleep decreased during the first 2 hours. These data suggest that transient increase of DA in the BLA during non-REM sleep might be crucial for triggering the transition to REM sleep. And there have been several reports suggesting a possible involvement of D2 receptors in the regulation of REM sleep. They are pharmacological study. And these studies imply that the DRD2 may play a role in the regulation of REM sleep. So this led us hypothesize that transient DA peaks act on DRD2 positive neurons, dopamine D2 receptor positive neurons in the BLA to initiate REM sleep. To investigate the effect of DA dopamine on DRD2 neurons in vitro, we conducted 4 cell recordings from DRD cells in the brain slice. After expressing SSFO, in BTD neurons, we prepared brain slices and subjected them to whole cell recordings. Excitation of DA fibers by application of light induced hyperpolarization in DRD2 positive cells, indicating that DA dopamine inhibited DRD2 neurons in the BLA. Notably, DRD2 neurons displayed very long-lasting hyperpolarization after light stimulation, highlighting that even though DA peak was very transient, it caused a very long-lasting inhibitory effect on DRD2 cells in the BLA. So next, we tried to mimic the hyperpolarization by directly inhibit DRD2 cells in the BLA. So for that purpose, we expressed BLWO in the DRD2 cells in the DRD2 Cree mice by applying AB vector, Cree-dependent AB vector, and we prepared slides preparations and subjected them to whole cell recordings. And we found that optogenetic inhibition, we are applying blue light to inhibit these cells through BLWO, and this manipulation caused hyperpolarization in a similar time course of that induced by excitation of the DA fibers. So given that DA induced hyperpolarization in the DRD2 neuron, the BLA, presumably the activation of GI proteins and the GAG channel, we examined direct optogenetic inhibition of these cells by activating GI-mediated pathway. So as I already said, we expressed BLWO in DRD2 cells in the BLA by injecting AV, DIO, BLWO in the BLA of DRD2 cell, DRD2 Cree mice. And as I showed you previously, the slice preparation studies with slice preparations show that shining a light causes hyperpolarization of these cells, similar to that observed when DA fibers are stimulated. Then we tried to examine the effect of this manipulation in vivo. We implanted optic fibers bilaterally in the BLA in DRD2 mice expressing BLWO in the BLA and optogenetically inhibited DRD2 cells in the BLA while monitoring EEG and EMG. The inhibition of DRD2 neurons in the BLA during non-REM sleep triggered non-REM to REM transition. REM sleep began about 19 seconds after the simulation, which was significantly shorter than that of control, expressing only EGFP. The the application of light inhibition of these DRD cells every 30 minutes, commencing at Zeitgeber time 8 and concluding at ZT11, robustly augmented the quantity, time of REM sleep, while concurrently reducing non-REM sleep. The EEG power spectrum during each state was not influenced by the optogenetic manipulation. The duration of non-REM sleep was shortened as a result of earlier termination of non-REM sleep episodes caused by the optogenetic inhibition of DRD2 cells. And force expression was observed after the force expression is observed in the cellular population surrounding YFP expressing cells, which means these cells are DRD positive cells, but not within the DRD2 positive cells themselves, indicating that the inhibition of DRD2 cells in the BRA resulted in the disinhibition of adjacent neurons in the basolateral amygdala. And chemogenetic experimentation further corroborated the significance of DRD2 neurons within the basolateral amygdala in regulating REM sleep. I mean, we expressed inhibitory direct receptor HM4DI in DRD2 neuron in the BLA of DRD2 Cre mice. And intraperitoneal injection of CNO augmented REM sleep while concurrently decreasing duration of non-REM sleep without affecting EEG power spectrum of each state. And we observed increment of host-positive neurons in both BLA and central nucleus of the amygdala, thereby confirming the disinhibition of the amygdala caused by the manipulation. The activity-dependent genetic labeling technique, named TRAP2, was employed to label activated neurons following the activation of dopamine fibers in the BLA. The HM3-DKM3 construct was utilized as an axonal tracer here. The regions containing M-cherry positive fibers were identified here. The projections were discovered in mesopontine junction regions known to be involved in regulation of REM sleep, including the sub-C or SLD, and dopamine signaling. This suggests that dopamine signaling in the amygdala inhibits basolateral amygdala, thereby promoting innervation to the regions within the brainstem regions implicated in the regulation of REM sleep. And we recently found that CNO administration to these mice robustly increased amount of REM sleep. Unfortunately, I haven't brought the data, which was unpublished. I guess Emmy might have data. Please recall the information regarding narcolepsy and cataplexy, and that cataplexy is a pathological manifestation of REM atonia during wakefulness. So since cataplexy is thought to be a pathological intrusion of REM sleep or REM sleep factors into the wakefulness, we next examine whether the signaling in BLA is also involved in emergence of cataplexy using narcoleptic model mice. We employed a GRAB-DA sensor in the BLA of narcoleptic orexin ataxin 3 mice, in which orexin neurons are genetically ablated, and installed an optical fiber within the BLA to assess fluorescence in order to evaluate a correlation between DA release in the BLA and cataplexy. To elicit greater number of cataplexy episodes, the mice were provided with chocolate as a dietary stimulus. Observations revealed transient elevation of DA during chocolate consumption. Emmy now put chocolate in the cage. Here, the mouse recognized the chocolate and sniffing. And as you can see, the DA level in the BLA was increased. This is a typical trace, which is in contrast, in the wild type mice, the dopamine levels didn't exhibit such a significant increase upon consuming chocolate. This is summary. So we observed a transient increase of DA level during chocolate consumption, which was preceded by cataplexy-like events. The elevation of DA levels in BLA during chocolate feeding in orexin ataxin 3 mice was found to be significantly greater than that observed in control wild-type mice. However, in contrast, the magnitude of the increase in DL level and in the nucleus accumbens during chocolate consumption was found to be almost comparable in between narcoleptic and control mice. Next, in order to ascertain the causative relationship between the increase in DL levels in the BLA and the cataplexy, we utilized optogenetics to selectively activate the BTA neurons during wakefulness. We again used SSFO. SSFO was expressed in BTA dopamine neurons of that iris mice, and bilateral optical fibers are implanted in the BTA for optogenetic stimulation. Following optogenetic, now we applied right. Following optogenetic stimulation, the mice displayed cataplexy-like behavioral arrest. The application of laser stimulation for 1 second resulted in transient increase of dopamine level in the basolateral amygdala in that iris mice, which displayed a pattern similar to that observed during chocolate feeding in narcoleptic mice preceding cataplexy. Notably, this increase in dopamine levels in BLA was accompanied by an abrupt behavioral arrest resembling cataplexy, despite the fact that these mice were not narcoleptic, not orexin deficient mice. Subsequently, we evaluated the effect of optogenetically stimulating the BTA fibers in various brain regions, including the BLA, nucleus accumbens, medial prefrontal cortex, nucleus accumbens, and lateral hypothalamic area. But excitation of these fibers during wakefulness resulted in cataplexy only when the optic fibers were placed specifically in the BLA. We never found a cataplexy-like episode when optic fibers are found to be in these other regions. Subsequently, we optogenetic inhibition. We tried to optogenetically inhibit dopamine fibers in the BLA. And optogenetic inhibition of dopamine fibers in the BLA completely abolished the cataplexy-like episode triggered by chocolate in the orexin ataxin 3 mice. This is the total time of cataplexy and number of cataplexy. We never saw the episode of cataplexy when we inhibited dopamine fibers in the BLA. So given the observed differences in dopamine dynamics across the sleep-wakefulness cycle and the effect of optogenetic stimulation of DA fibers between the BLA and the nucleus accumbens, it is likely that distinct subpopulation of dopamine neurons in the ventral tegmental area send projections to these regions. In fact, retrograde tracing with retro beads injected into the BLA or nuclear accumbens in wild-type mice revealed a lack of double positive cells in the BTA, suggesting that the subpopulation of dopamine-releasing BTA neurons that project to the BLA and nuclear accumbens are distinct. Specifically, dopamine-releasing BTA neurons that innervate BLA were found to be localized in the dorsal regions of the BTA, while those that project to the nucleus accumbens were found to be in the ventral region of the BTA. This is a summary. Our findings indicate that during non-REM sleep, dopamine levels in the BLA periodically increase, which act on DRD2 expressing neurons to terminate non-REM sleep and initiate REM sleep. Suppression of this mechanism led to a significant inhibition of REM sleep, whereas mimicking the increase in DA levels in the BLA-induced REM sleep. This reveals the previously unknown and definitive mechanism controlling REM sleep. Additionally, our research also suggests that this mechanism also plays a role in the occurrence of cataplexy in narcoleptic animals. Specifically, in narcoleptics, DL levels in the BLA increase to a higher level in response to positive emotional stimuli, suggesting that orexin might act as a gatekeeper of DL levels in the BLA during wakefulness to inhibit cataplexy. Last but not least, I'd like to express my gratitude to the member of our laboratory and collaborators who have contributed to this study. In particular, I'd like to extend my heartfelt appreciation to Emi Hasegawa, who is attending here, and Shuntaro Uchida for their invaluable contributions. And I'm deeply grateful to Dr. Yuron Lee for kindly providing us with valuable blood sensors. Thank you very much for your attention. I'm ending my presentation. Thank you.
Thanks a lot, Takeshi. So I'm very glad to have you here online, and it was a great talk. So and the idea also with this webinar is to be a bit lively and to be able to discuss together more like a round table. So I'm glad we solved the technical issue and we can go on. Maybe I will start to ask. I see Amy now is here, so hi Amy. I'm glad to see you too. So of course, when we see you thank a lot of people at the end of your talk, and one maybe general question before we start to go into the detail and of what you did and your results is, okay, this is a big paper published in a famous journal, and I see you have a huge amount of data provided for your demonstration. So this is at the moment necessary to have this kind of publication. So a more general question, maybe quite unusual for you, would be, okay, how much time you needed to complete this huge project, right? How many years and how many people were involved in this huge effort and amazing set of data?
I remember Amy started this project shortly after she joined our team in Tsukuba University. So I guess it was 19 no, 2016, right?
Okay.
Yeah, right.
20. So it took almost 5 or 6 years, I think.
Yeah.
They're almost all of these data are done by high effort, I think, I guess. Almost 80% of these data I presented today were done by Emmy, I guess.
Okay.
Well,
Yeah, that
of course, there are many people who helped her experiment.
okay. Yeah, I believe that's important to tell people right here listening to the webinar how you proceed to make such nice demonstration, and because I mean that's a way people needs to learn to construct such amazing set of data. If I go now, the question are open, so we will start to get the question from the audience, but I might start like a more general comment about now your findings. I would say that, as you know, I devoted my life to paradoxical sleep. I say not REM sleep.
Yeah, I know.
It's the same, you know. But you know, something amazing to me is that when I saw your paper on basolateral amygdala, of course, I went into it in detail, and I look at everything, and I found it fascinating, well designed, and all with a very nice number of amazing experiments. So I'm very convinced by your demonstration. I have to say, and it really I didn't find like some technical explanation could tell us why you found something completely unexpected. So that's the first point. So then I try to understand how I can explain your results when taking into account the previous knowledge on the field, because of course it's quite surprising that a basolateral amygdala, which is more known as involved in emotional regulation and memory. So I was very surprised that this structure, and moreover, of course, the dopamine input on this structure. So here you have quite crazy stuff, because not only basolateral amygdala is not a structure involved in REM classically. So first, and second, dopamine, of course, is not directly involved in REM generation, right? It's more like wake recently a system. So that here you have two very surprising results. And I did try to understand how you can bring this together with what we know previously on REM sleep. So I would like to ask you here this question. So how do you explain that you would need dopamine increase previously to REM in basolateral amygdala, which is emotional structure to induce REM sleep?
Actually, initially we intended to examine the involvement of dopamine in the pathophysiology of cataplexy, because as you know, cataplexy is usually induced by positive emotion. So we postulated that dopamine might be playing some roles in induction of cataplexy. So I asked Amy to perform optogenetic excitation manipulation of dopamine neurons in the BTA by channelrhodopsin 2 or some other opsins. And incidentally, she found that optogenetic excitation of BTA dopamine neurons in some conditions can induce cataplexy-like behavioral arrest in mice. And actually, we found that excitation of the neuron in the BTA caused complete atonia, while EEG shows some resemblance to the wakefulness or REM sleep. So we postulated dopamine might be playing some role in induction of cataplexy. And furthermore, we knew that there are several dopamine sensors, GPCR-based dopamine sensors, and Euron Lee came to our institute, and we discussed with him about using the dopamine sensors to monitor the dopamine level in several brain regions. And we actually measured dopamine levels in several brain regions during measuring EEG-EMG to monitor the sleep and wakefulness states. And Amy found that dopamine peaks were found to be just prior to non-REM to REM sleep transition. So we hypothesized that dopamine peak within the BLA might trigger the REM sleep. When these peaks are caused, elicited during the non-REM sleep, that might result in the termination of non-REM sleep and the initiation of REM sleep. And when these peaks occurred during wakefulness, that might cause the occurrence of cataplexy. So we concluded that the dopamine peak in the BLA might be very important. And as you know, amygdala is well known to be activated during REM sleep. So this dopamine might be, we guess this dopamine might be also important to activate dopamine regions. So in conclusion, activation of amygdala regions by dopamine peak might be playing very important role in REM sleep regulation. And we actually found that activated neurons, I mean, DA activated neurons in the BLA send abundant projections to the SLD or other brainstem regions that are implicated in the REM sleep regulation. So we concluded.
Yeah. That's interesting.
This conclusion.
Yeah, that's very nice comment, Takeshi. I like, and when I read your paper, I read and I saw REM and cataplexy. I thought, okay, they wanted to study cataplexy. It was not the same to look at a REM sleep.
Actually, we initially thought to examine the pathophysiology of cataplexy, and we examined the possible involvement of dopamine in the pathophysiology of cataplexy. And in a separate study, we found that the neuronal pathways involving in the REM atonia and cataplexy are identical. So we think cataplexy and REM sleep might be caused by very similar mechanisms. So during doing the experiments to reveal the dopamine role in the cataplexy emergence, we also examined its role in REM sleep regulation.
Yes. No, that makes sense this way, right? Because I mean, there was no reason to go to look at this dopamine in basolateral amygdala for REM sleep without, arriving there because you had these cataplexy ideas before. So now it's much more clear for me. Maybe we can open the discussion to the people attending the meeting. So I don't know if technically I see that Rick Wassing, you ask a question. I don't know if you can really we can activate your video so that you can ask your question or I need to summarize it. I don't know if Takeshi, if you see the question of Rick, he said that there was there is an increase of dopamine from VTA to BLA just prior to REM. But also inhibition of dopaminergic neurons in the BLA induced REM sleep. So does that mean that the dopaminergic neurons in BLA have dopamine receptors, but that hyperpolarize these neurons? So that's indeed what I understood from your data, right? I don't know if you want to answer to that. For me, I understood indeed that you have inhibition of the terminal, the excitation of dopaminergic neuron terminals in the BLA. So inhibits, and that's something I wanted to ask you. They inhibit GABAergic neurons, right, bearing D2 receptors, because you don't directly say that
these DRD2 cells are GABAergic, I guess.
Thank you. Yeah, they unmuted me, so I can speak up. But that's
okay.
That's good.
Great.
Good to get confirmed. Thank you.
So go on.
Yeah, no, I mean, you've answered the question that indeed the DA excites GABAergic neurons, which then indeed inhibits the BLA, which is what my question was about. Thank you.
Okay, so I have a comment, by the way, Amy and Takeshi, about the pathway, right? It was, I was a bit confused and I was missing some link. And I like to make pathways, to have drawings with pathway. I'm a specialist to draw that kind of stuff. So I understand that dopamine neurons inhibit GABA neurons with D2 receptors. So it means that these GABA neurons normally inhibit glutamate neurons, right, in the BLA. And so these glutamatergic neurons are disinhibited. And then how do you go from there to the brainstem? That's my first idea, because there is no projection from the BLA to the brainstem, to my knowledge. I'm not aware of any such projection. So I wonder if you believe that these glutamatergic neurons, they project to GABA neurons, and they excite GABA descending neurons in the central amygdala, and that's the way you could.
Right.
Okay.
Yeah, these, as you suggested, the dopamine inhibit GABAergic interneurons, which express DRD2. So inhibition of these interneurons resulted in disinhibition of glutamatergic neurons in the BLA. That some of these cells directly send projections to the brainstem regions, and some are sending projections to central amygdala, CEA.
Yeah.
And CEA, we found that CEA neurons are also excited because false positive cells in the CEA were also increased after the manipulation. So CEA regions are also excited by the dopamine peak. So, and these excited cells, we identified these excited neurons by TRAP2 method, and we found these cells are actually sending projections to SLD or other brainstem regions. So CEA SLD or BLA SLD pathway might be playing a role in regulation of REM sleep.
Yeah, thanks a lot. Takeshi, for this explanation. So that it will be important, of course, to define the descending pathway in detail in the future. I believe we have a question here. We have Joyce, who is visible online here. So please, Joyce, you have a question? Can you ask your question?
Sure. I'm always trying to translate these studies into practical use for patients. And when I saw the presentation, I was thinking about how many of our patients use melatonin. And I remember melatonin has an interaction with dopamine. And I wonder if there's any data, if the wonderful results that were very elegantly shown, if those results might change when the melatonin background changes during the night, but also in the use of pharmacological melatonin.
Yeah, we haven't examined the effect of melatonin in this system.
Not yet, probably.
Not yet. But in mice are nocturnal, but melatonin level is increased during the night, I guess. So there might be some difference between the nocturnal and diurnal animals.
Thanks a lot. Maybe we can proceed. With other question. So I see that Antoine Adamantidis and Mattia Aimé are also online, and they are also asking a question about the latency to REM onset, and that's a long latency. When you stimulate, you see that you have several seconds, right? 20-second latency, something like this. And I don't know if Antoine wants to directly
almost
comment.
2 minutes.
Almost
2 minutes.
Yeah, so it's quite long latency, and we wonder. Antoine?
Yeah, I'm here. I'm online. I don't have the video, but I can
go on.
Join.
It's nice to get you online. Yeah.
Emi Takeshi, this is a really nice paper, really interesting set of data.
Thank you.
And yeah, the question is about your interpretation of the long latencies. This is something we see in many circuits. And your suggestion that the VTA, dopamine to BLA and to other REM sleep circuit might be at play here is of course what we all think. Now as you just discussed with Pierre before, the connection between VTA, BLA, and then BLA to REM center is at least 2 synapses, maybe more. So how do you interpret the long latency? Is there a build-up? Yeah, is there a build-up? Is there
basically dopamine signaling is a kind of slow process. The metabotropic neurotransmission. So there are several steps in dopamine signaling. So it might take several minutes, I guess. So dopamine release and binding to the receptors and activation of G proteins, and then second messengers generated, or cyclic AMP levels decreased, and GAC channels and beta gamma subunit activated GAC channels. So there are many steps. Dopamine signaling itself is very slow process. So I guess this can explain why the latency from the activation of dopamine fibers to the REM sleep initiation might.
And then the dopamine that you show with the sensor is increasing right before REM sleep, so that would make sense. But it decreased quite quickly after. So really, yes, it fits with the idea that it could signal onset. So based on what you just said, then I would suggest that it's more downstream than of the BLA, right? That there is some delay.
Yeah.
If dopamine transient is
And
short.
This is a multi-synaptic pathway, I guess. So, and we, as I showed, the dopamine peak itself was very transient, but the inhibition of BLA neurons by dopamine lasted several minutes. So this long-lasting nature might be important to stability of REM sleep, we guess.
Yeah. Interesting. Thank you.
Yeah, thanks a lot, Antoine. Glad to get you online with the question, and it's a lively discussion. So maybe I have a few comments, and we need to move to the next speaker, Takeshi, just to talk about an interpretation of your data we might have. Indeed, so when I saw your results, and that's something I wrote a comment on it, but I don't know if it will be published sooner or later at the end, but I was invited to make some comments. And I thought, you know that, of course, this structure, basolateral amygdala, is involved in emotional regulation, emotional memory, and of course it has also projection to prefrontal cortex and so on. So I also wonder whether we could explain your results rather than saying that it's directly, I would say, playing with the REM generating network, like with a projection to the brainstem, that in fact part of your effect could be due could be indirectly due to a modification in the function of the state of REM sleep. Because imagine that this structure, I mean, we know that this structure is involved in emotional memory, and there is a strong link on role and function of REM sleep in emotional regulation.
Right.
So in a way, you play with emotional regulation when you stimulate and inhibit basolateral amygdala neurons, right? It's like you tell the animal or you tell the brain of the animal that emotionally something is happening. And I was wondering, in fact, if your result could also not be explained by such effect on the function of the state, you know, like if you in fact stimulate the function of REM sleep or inhibit the function of REM sleep in the regulation, in the emotional regulation, you could also obtain what you see, like an increase or a decrease in the quantity of REM, because you need, let's say, emotionally you have something to cope with, then we know you increase REM, and conversely, maybe if you are fine emotionally, you need less REM. So I wonder what you think about this on other pathway, in fact, than those to the brainstem.
Yeah, that's a very tough question. As you know, basically, limbic system is activated during REM sleep. So, but no one knows what they are doing during REM sleep. So amygdala and the hippocampus are also activated during REM sleep. So some researchers hypothesize that some memory-related mechanism might be working during REM sleep, but the dysfunction no limbic system during REM sleep not solved, I guess. But so this mechanism, I mean, the dopamine increase in the BLA might be one of the possible mechanisms that activate amygdala and other limbic system. I don't know, but other limbic system might be also activated by this mechanism in the downstream of amygdala. So the activation of limbic system might be one of the hallmark of REM sleeps. So this might be some kind of keys to know the role of physiological role of REM sleep in the future, I guess.
Yeah, thanks a lot. Thanks a lot, Takeshi. I believe, yeah, you open a new door. I mean, in the history of REM sleep with your paper. So I believe it's very fascinating, and it will be followed by amazing new ideas and findings. So thanks a lot for your communication, the discussion. I was so excited, and we need now to move on to the
thank you for this opportunity.
To the next speaker. Thanks a lot. It's
I found that there are many questions, but the direct emails is welcome. So please send me emails if you.
Or you can you know that you can answer in the chat here directly to people who ask a question now. So thanks a lot again, but I would say that we need to move to the next speaker. So if Xiao, you can now start your talk and your presentation. And thanks a lot, Emi and Takeshi. For your communications. I don't know if you are ready to start your online presentation. Okay, I leave you the floor. Thanks a lot.
Okay, so can you hear me?
Yeah, everything is fine.
Well, thanks for the invitation by Dr. Lupi. And it's a pleasure to share some of our latest research outputs in this roundtable meeting with further discussion. So Professor Bill Whiston and me today will talk about how we define ventral tegmental area, VTA, in the midbrain as a critical brain area for sleep-wake regulation, in addition to how stress is engaged in this circuitry to produce restorative sleep that helps the mental processing of stressful events. So sleep is an essential physiological process. Most adults require about 8 hours of sleep at night. Older adults tend to sleep for shorter periods. And sleep is of the brain, by the brain, and for the brain, and is certainly beneficial and restorative. Sleep is essential to our survival. It can improve our learning ability and consolidate our memory. Sleep loss can reduce the connectivity of the brain. For example, sleep deprivation reduces connectivity between amygdala and prefrontal cortex, hippocampus, and other cortical areas. Sleep helps to clear metabolic waste. So here shows the CSF circulation is much more vigorous during sleep, which can help brain clearance. And sleep can balance the production and the release of hormones. So if we don't get enough sleep, we might get obesity due to a disrupted hormone level. But the most important issue is safety. Insufficient sleep causes microsleep during driving, which causes car accidents. It has been shown that 24 hours of enhanced wakefulness has a greater detrimental effect on driving ability than consuming the legal limit for alcohol. Here is the test. That's the red line represents the 24 hours enhanced wakefulness. The performance very bad. So we use animal models to study sleep and its related behaviors. So here we summarize the proposed circuits of wakefulness and sleep in mice. Several pathways have been demonstrated to promote waking. Monoaminergic cell groups drive arousal by producing noradrenaline, serotonin, dopamine, and histamine, which innervates with large cortical area, basal forebrain, hypothalamus, and many other brain areas. For example, the tubermalinary nucleus that we explored previously is the source of histamine in the brain. And during wake, histamine neurons excite neurons in the cortex, thalamus, and other arousal-promoting regions. So we use transgenic reporter mouse line to illustrate histamine cells in the hypothalamus. And most of the sleep-promoting cells release GABA to inhibit arousal-promoting elements. So recently, a few excitatory circuits have been identified to promote sleep, including substantial nigra and a very specific area in the midbrain. And we are particularly interested in the ALPO. This shows ALPO GABAergic and galaninergic neurons with GFP. And in particular to the VTA. This image shows the major cell populations in the VTA. Green represents GABA and dopamine and glutamate. But still, we do not know all the circuits that regulate vigilant state. Therefore, we used an unbiased chemogenetic approaches searching for glutamatergic neurons in the posterior hypothalamus and midbrain that could regulate vigilant state. So first, we expressed the excitatory HM3 receptor in VGRU2 neurons in progressively more defined locations. First, large volume injections induced 100% wakefulness for about 12 hours. A smaller AAV injection resulted in HM3DQ in the hypothalamus and also midbrain, produced continuously wakefulness for about 6 to 7 hours. And then we restricted the HM3DQ in other specific brain regions. Expression to the LH also produced extended wakefulness. By contrast, HM3DQ expression in the mammillary area and in Duncan nucleus had no effect to sleep-wake state. And of note, we expressed the HM3DQ in the VT area following CL injection. There was about 5 hours of 100% wakefulness. Of these brain areas, the VTA VGRU2 population that promotes wakefulness had not to our knowledge. So therefore, we decided to explore the VTA in depth. So VTA locates in the midbrain and is an important source for dopamine, glutamate, and GABA. The VTA has been intensively investigated for its regulation of rewards, addiction, and social behaviors. For example, dopamine neurons receive excitatory inputs from other brain areas and project to the prefrontal cortex to regulate aversion, reward via dopamine signaling. And BNST controls rewards learning aversion behavior through VTA by releasing neurotransmitters. Also, VTA GABA can regulate learning aversion. In addition, VTA glutamate neurons also control aversive-like behavior via the electrohambular or nuclear compound pathways. Of course, loss of dopamine neurons or VTA dysfunction has been implicated with depression, Parkinson's disease, or Alzheimer. These dopamine, GABA, and glutamate neurons control diverse behaviors, but are governed by vigilant state. As we found that activation of VTA VGRU2 positive cells promote waking, so we next decided to explore GABAergic population and their circuits for sleep-wake regulation. We first used the calcium indicator to determine the dynamic activity of VGRU2 VGAT population during natural sleep-wake period by delivering GCAM6 into the VGRU2 or VGAT creep mice. So as we can see, calcium signal was much higher during wakefulness and REM sleep compared with non-REM sleep in both wake group 2 and wake positive neurons, which suggests wake group 2 and wake neurons are primarily wake and REM active. So we next conducted chemogenetics to explore whether wake neurons can induce sleep or wakefulness. So we expressed excitatory HM3 DQ receptors in VTA wake group 2 cells, and we found chemogenetic activation of VGAT population in the VTA produces long-lasting non-REM sleep for about 7 hours. We then undertook channel rhodopsin-based tracing to look at the downstream targets. As we can see, there's a dense long projection of VTA wake group 2 cells to the lateral hypothalamus, LHB, and the forebrain, including nuclear accumbens. However, for VGAT neurons, terminals were found locally in the VTA, but also in the LH area. We then placed optical fiber above those targets, and the optical activation of VTA wake group 2 to the LH and nuclear accumbens induces wakefulness from non-REM sleep, whereas activation of VTA VGAT to LH pathway promotes sleep from waking. So thus we have demonstrated there are circuits that control sleep and waking. So this previous work suggests that VTA has a powerful role in regulating sleep and wakefulness. GABAergic, dopaminergic, and glutamatergic cells are active during wake and REM sleep. During natural non-REM sleep, all types of cells were silent or less active. Artificially, activation of wake group 2 neurons promoted wakefulness through LH and nuclear accumbens pathways, but activation of VGAT neurons inhibits local dopamine and wake group 2 neurons, as well as orexin cells in the LH that we explored before, and subsequently induce deep long REM sleep. And we hypothesized that physiologically, VTA wake group 2 and dopamine neurons sustain wakefulness by exciting the nuclear accumbens and orexin cells in the LH, and the VTA VGAT neurons provide an ongoing inhibitory tone onto the wake group 2 and dopamine neurons, and to maintain the network function. So given the hub-like nature of those GABA cells for vigilant state control, it is still unknown that how these neurons are controlled under physiological conditions. VTA integrates internal and external information to guide diverse behaviors via distinctive pathways. In addition, VTA also remarkably responds to threats and stressors. Therefore, we propose that VTA framework may provide a link of stress to sleep-wake regulation. In our follow-up work, we extended our study by investigating how VTA processes stressor to guide behaviors. Stress is commonly defined as a state of threatened or perceived homeostasis, which requires activation of complex range of responses. Optimal basal level is essential for successful performance and appropriate social interactions, but excessive leads to a host of pathological conditions. And stress correlates with mental disorders such as anxiety and depression. It also can cause cardiovascular disease and impair our immune function. It can reduce hair loss, white hair, and skin problems. Animal models of stress has been widely used to investigate stress responses, stress-related behaviors, and psychiatry, mental disorder. So those stresses procedure include restraint, chronic mild stress, and also social defeat stress. Social defeat stress based on resident intruder protocol is a form of psychosocial stress that mimics bullying in humans that causes anxiety and depression. Exposure to environmental, physical, or psychological stresses leads to activation of stress response. which includes a complex interaction between components of the central nervous system and peripheral systems, including the endocrine, immuno, and the cardiovascular systems. For example, a model of network responding to psychosocial stress, associative stress, multiple inputs from the cortical limbic circuits were integrated to process stress and emotion. So DMH receives glutamatergic signals and they activate paraventricular hypothalamus endocrine neurons to stimulate stress hormone release. The excitatory group of DMH neurons also control different effectors, drive sympathetic outflows to the brown adipose tissue, heart, and blood vessels through different pathways. Another subset of DMH neurons also drives avoidance behavior. In normal physiology, three structures modulate the response to stress. The classic HPA pathway. The HPA axis plays a central role in regulation of adrenal hormones that helps to preserve or restore homeostasis. For example, cortisol in human and corticosterone in mice were elevated in response to stress that caused an increase of heart rate, muscle strength for a flight or fight response. And also, for example, we observed an increase of corticosterone concentration in blood after associative stress in mice. So most stressors promote insomnia, and this is people's common perception. Yet some psychosocial stressors such as social defeat stress can induce sleep. Some studies discovered that social defeat stress in mice and rats is the most reliable form of stress that induces non-REM sleep for continuously about 5 to 6 hours. But REM sleep differs between different stress protocols. However, the neurobiologic mechanism of how social stress induces sleep is still unclear. Because VTA neurons are activated by stressful and aversive stimuli, and we have shown that VTA has a very powerful role in sleep-wake control. So we consider this pathway might allow stress to induce sleep. So to begin with our study, we determined the sleep-wake architecture in mice after social defeat stress. So here we used a 4-episode protocol of social defeat stress in an hour, and then the mice were placed back in home cages. Their sleep-wake states were recorded, consistent with our reports. Social defeat stress had a substantial effect on promoting non-REM sleep for continuously 5 to 6 hours. And of note, our stress protocol also promoted REM sleep for about 5 hours. So after confirming that social stress can induce sleep, we next explore the potential functions of sleep after social defeat stress. First, it has been shown, and also we observed, that social defeat stress causes anxiety-like behaviors in mice as measured by elevated plasmase and open field assays. However, for mice allowed sufficient recovery sleep after social defeat stress in their home cages, anxiety-like behaviors were rapidly reduced. However, if mild sleep deprivation over 4 hours took place immediately after social defeat stress, the mice remained in an anxious state, suggesting sleep after stress alleviates stress-induced anxiety. So we next aimed to identify the elements activated by the stressors that induce this sleep. So first, we mapped the C4 positive cells activated by social defeat stress throughout the whole brain. After social defeat stress, C-force was elevated in few classic stress detecting areas, including APO, BNST, PVH, LH, and also other PVT and other brain areas. These brain regions contain distinct populations that can regulate sleep-wake states. Notably, C-force was also elevated in the VTA. And given the heterogeneity of the cell populations in the VTA, we first characterized the stress-activated cells with defined cell types. And we found a majority of the C-force positive cells in the VTA activated by stress are GABA neurons. So we expressed M-cherry in VGAT positive cells and double labeled with C-force, as we can see here. And also, we used a health indicator to determine the acute response using GCAM6. We expressed GCAM6 in VTA VGAT neurons, and these cells rapidly and strongly responded when mice experienced an attack during social defeat stress. We also looked at the long-term effect of social defeat stress on VGAT population activity. So very interesting, we found the baseline activity of these cells were elevated during the stress period. And the average population activity was enhanced after social defeat and persisted for about 5 to 6 hours, which suggests stress remodels the activity of those VGAT populations in the VTA, and the enhanced activity following stress allows these cells to maintain their function of sleep induction. So having noted that social defeat stress induces sleep, and subsets of VTA VGAT neurons were excited by stressor, our follow-up work explored how VTA VGAT neurons link stress and sleep induction. To do this, first, we need to capture the key cells, as they are only a subset of total VTA GABA cells. So we undertook a C-force dependent activity tagging approach with HM3 DQ or other genetic tools to test if these stress sensing cells in the VTA could induce sleep. So we generated this AAV-based activity tagging approach in our lab previously with two constructs, the AAV C-force TTA and the AAV TRE response element carrying HM3DQ or HM4 and DI. So this shows we co-inject these AAV constructs into the apple, and then we use dogs to inhibit the system, and then remove the dogs, and the mice were given sleep deprivation or recovery sleep, which can acutely labor the cells activated by sleep deprivation and recovery sleep. And then we can use CNO or other small molecules to manipulate the activity of these neurons. So here we undertook this C-force dependent tagging system with HM3 DQ and cherry to test if these stress sensing cells in the VTA could induce sleep. So mice experienced social defeat while the GABA cells were activity tagged. So this image shows stress labored VTA GABA neurons. You can see located in a specific area in the in the middle of the VTA. We then artificially reactivated the tagged VTA VGAT neurons using CNO, and then we found chemogenetic reactivation of these specific stress tagged cells increases both long REM and REM sleep. So a small population of VTA GABA neurons respond to stress is sufficient to induce non-REM and REM sleep. And also, we can conclude that reactivation of social stress-activated subsets of GABA cells generated an identical sleep pattern, sleep induction to social diffuse stress induced, as we can see here, which suggests reactivation of tagged neurons recapitulated social diffuse stress induced non-REM and REM sleep. And to examine the necessity of those subsets for stress-induced sleep, we chemogenetically inhibit social diffuse stress tagged GABA cells using C-force-dependent HM4DI. So the mice were co-expressed with the activity tagging chest genes, and the mice were subject to first stress episode to allow cells to become tagged with HM4DI, and then given CNO to inhibit the tagged neurons. And the mice were subsequently challenged with a second bout of stress episode. As we can see here, social diffuse stress-induced sleep was diminished after chemogenetically inhibition of tagged VTA-VGAT neurons, suggesting these cells are necessary for stress-induced sleep. And next, we investigated the circuitry linking social stress and VTA-VGAT induced sleep. VTA-VGAT cells extensively project to other brain areas, including prefrontal cortex, lateral hypothalamus, lateral hypannular, central amygdala, and lateral hypothalamus. So we used a calcium indicator selectively in VTA-VGAT neurons, and then used fiber photometry to measure how the VTA-VGAT terminals in different locations responded to stress. And we found only the terminals of the GABA cells projecting to the lateral hypothalamus had increased calcium signals during social diffuse stress. So which means this the VTA to LH pathway might transmit social diffuse stress. So to determine the function of VTA-VGAT to LH pathway activated by stress on sleep, here we used a three-way intersectional genetics with a retro AAV and theta response to create dependent FLIPO into the LH together with injection of C-force TTA and FLIPO dependent HM3DQ into the VTA of VGAT neuron. So following intersectional activity tagging, which allows the LH projecting GABA cells to be tagged during social diffuse stress. So those cells were tagged during social diffuse stress, but only project to the LH. And we found chemogenetic activation, reactivation of LH projecting VTA-VGAT cells promoted both non-REM and REM sleep. And we also investigated the identity and activity of inputs that were relevant for social stress. For this, we used a novel RABI system combined with C-force-based activity mapping. So we expressed the VTA-VGAT neurons with these rabies components following the injection of rabies virus into the VTA. Few days later, the animals were given social diffuse stress. We conducted a whole brain mapping of rabies labeled presynaptic inputs and stress-activated C-force expression. And we found only ALPO, PVH, and PAG had overlap with C-force positive cells and the rabies labeled presynaptic inputs, suggesting VTA-VGAT neurons receive stress-driven inputs mostly from the ALPO, PVH, and PAG. And also, given that GABAergic in the VTA are heterogeneous and only a subset of VTA-VGAT cells responded to social diffuse stress, so we looked for subtypes of VTA-GABA cells responsible for social diffuse stress-induced sleep. And first, we examined by single cell qPCR the molecular identities of those stress-tagged cells, and we found a large proportion cells expressed VGAT somatostatin marker, and we further characterized activity cells also using the reporter mouse line. We found nearly 40% of the VTA somatostatin positive neurons expressed C4s after social diffuse stress. We also determined the activity of individual subtypes responding to stress. So here it shows using fiber photometry. Here shows the VTA somatostatin populations responded transiently to social diffuse stress. And this SST subtype had persistent activation after social diffuse stress with enhanced activity for a few hours, which is identical to the VTA-GABA populations. We also measure the spontaneous activities of SST subtype across brain states. From casting photometry, found that SST neurons were primarily active during spontaneous non-REM and REM sleep. And this is very interesting because we previously found that the population activity of total VGAT positive cells were weak and REM active. So here we might discover a subset of sleep active GABA cells in the VTA. And these cells could be the key cells for sleep induction. We next examined directly whether SST neurons are necessary for stress-induced sleep. So we used a genetic ablation approach to specifically deplete VTA and SST neurons. And we found enlisting of VTA-SST neurons decreased baseline sleep. But when CAS-based mice were challenged with social diffuse stress, the stress-induced non-REM sleep and the REM sleep was abolished, suggesting these cells are necessary for stress-induced sleep. Given the proposed restorative function of sleep after social diffuse stress, we also explored whether this function was linked to VTA-SST neurons. After social diffuse stress, mice showed anxiety-like behaviors, but when SST neurons were ablated, the SST-induced anxiety-like behaviors still existed. However, if the GFP mice here, as you can see, the mice had a sufficient recovery sleep, and the anxiety-like behaviors were reduced to baseline. Here, the CAS-based mice, lacking SDS-induced sleep, remained in an anxious state, suggesting VTA-SST cells are required for the anxiolytic effects. However, we still do not know how does VTA-SST circuit reduce anxiety. So it could be stress-induced sleep, because as we can see, with sufficient sleep, the anxiety-like behaviors were reduced. And if sleep deprivation protocol, and the mice remain in an anxious state. So one possibility is stress-induced sleep helps to reduce the anxiety-like behavior. And another possibility is that the activation of these VTA circuits, because when we use genetic ablation or HM4 DI to manipulate the function of VTA-SST neurons, and actually also deactivated or ablated and the normal function of those cells. So therefore, we designed an experiment by expressing HM3DQ in SST cells to manipulate their activity. So after social defeat stress, the mice were followed by a sleep deprivation protocol with a saline or saline injection. So we found the animals remain in anxious state after saline injection. However, during sleep deprivation procedure after social defeat stress, while the mice were awake, activation of somatostatin cells did not reduce anxiety, suggesting that the anxiolytic effects require stress-induced sleep rather than VTA SST cell activation. Despite the restorative function of sleep to reduce anxiety, we also observed that for mice allowed sufficient home cage recovery sleep, the rise of corticosterone concentrations as a marker of stress response returned to baseline over the 6 minutes. If mild sleep deprivation occurred immediately after social defeat stress, the corticosterone concentrations remained elevated. So we substantially explored how SST cell is engaged in this process. Because corticosterone was produced by CRF, cortical releasing factor, we next directly determine the CRF production in the PVN. So our collaborator, Professor Yulong at Peking University, kindly offered us a novel and genetically encoded CRF sensor, which allows us to determine the CRF level in the brain. So we expressed the CRF sensor in the PVN and observed an increase CRF levels when mice responded to stress. So we used this sensor to investigate the connection between VTA and the PVN. So how does VTA and SST neurons regulate corticosterone production? So we found that VTA SST neurons expressing HM3DQ sent numerous positive axons to the paraventricular nucleus. So we next use this sensor to determine the dynamics of the CRF release around the PVN area. So we found after social defeat stress, there was a large increase in the CRF concentration. However, chemogenetic activation of those VTA SST neurons prevent this increase, as you can see here. And by contrast, chemogenetic inhibition of those SST neurons further elevated the stress-induced CRF concentrations. Thereby, we can see an increase of corticosterone levels after social defeat stress. So taken together, these results suggest that VTA SST neurons can innervate paraventricular hypothalamus to regulate CRF production. So in summary, so we discovered a subset of specialized neurons receive stress inputs from stress detecting brain regions. And these cells sense stress, and then their activity was enhanced persistently to induce sleep through the lateral hypothalamus pathway. And stress-induced sleep generated by this pathway mitigates stress-induced anxiety, and with innovations of the VTA to the PVN pathway, and also, of course, stress-induced restorative sleep together to alleviate corticosterone concentration in mice. So this specific circuit linking stress and sleep may provide a refined approach for treating stress-induced mental psychiatric disorders such as anxiety. So I would like to thank Professor William Whiston and Nick Franks at Imperial College, and all my colleagues. They contributed a lot to this project, and also our collaborators at Xijin Hospital in China with Hai Longdong and co-first author Guang Chaozhao. He has done a very hard working for this project. And also we have to thank Professor Yulong Li and Wang Huan for the generation of this CF sensor, and also Julia at Crick Institute for the VGAT photometry, and to thank everyone attending this meeting.
Thanks a lot, Zhao. It's really a second paper, amazing. You have been presenting here. It's also fascinating. I don't know if I thought at one point to relate the two papers, but maybe it's a bit difficult, but it's interesting that the two papers are looking at the VTA neurons, and that there is an effect on REM, and the two papers. But let's focus on of course first your data, and hi Bill, I'm glad also you are here to debate your paper. So I mean, for me it was also quite amazing to see how many experiments you did provide in this paper. I have to be honest with you, it's very difficult to read everything including all your supplementary data, because there are so many experiments there. So in a way, it's difficult to. It's like you have a life here, right? You have already like a lifelong study in your paper. So it's amazing. I found it quite also impressive that and also fascinating that you have this social defeat stress effect and inducing sleep. That's also something very interesting, because in my mind, you know, I was more on the idea. I mean, it's quite. I want you to I mean, maybe to explain a bit more about the relationship between stress and sleep, because for me it's not that homogeneous, right? Maybe you said that at one point, that depending on the type of stress, you might have different effect. And stress is a complex. It's not a single, like I would say, physiological. Stuff. I mean, you have different type of stress, and I remember in the lab of Michel Jouvet, Raymond Cespuglio, showing that, for example, if you restrain the animal in a tube for 1 hour and a half, we obtain at that time an increase of REM, a specific increase of REM, just following up during the restraint of the animal, the restraining of the animal. And I also discussed with other people saying that even social defeat in the same kind of experiment you did, they obtain wake rather than sleep, and so. And also the other stress we know are rather inducing insomnia, or so I want here. I believe it's important for us to discuss the impact on stress on sleep more generally, and whether it's, and also maybe I would like you to open your results to what's happening to humans and to some patients. So do you have an idea of application of your findings to humans? So that's the first question I had in mind.
I think in human, the perception is when we experience any stressors, actually the common phenomenon is stress promotes insomnia rather than sleep. But I think it may depend on the nature of the stressors, because I remember it has been shown that a person with a lighting, that is the natural disaster, a kind of stress that's make this person sleep better during the night. Probably depends on individual experience, but mostly stress in human causes insomnia rather than sleep. And actually this also applied to animal models, because most of the stressors also induce insomnia in mice rather than sleep. So only the social diffuse stress that has been shown the most reliable kind of stressors that induce sleep. But somehow that also depends on the habituate conditions after stress, because another group showed that if the mice were not in their home cages, the social diffuse stress also promotes microarousal that causes insomnia. And given the other stressors that you mentioned, the restraints or immobilization, that also induces sleep, but not immediately after stress. But that was during the sleep or wake period after 12 hours. So probably that's a kind of different mechanism rather than the observations that we found. The cells has been activated directly during stress and then induce sleep. I think that other peripheral or hormone or endocrine systems have been involved in the regulation of sleep and wake.
Just like to add something to that. Hello, everybody. So thanks, Xiao.
Hello.
Actually, I think I'm agreeing with Xiao's comments, but various people did write to me who were not sleep scientists, but were scientists from around the world, and said intense stress made them go to sleep. So I know that is a kind of an unusual thing, but and it's not scientific, it's anecdotal. But it is a very peculiar kind of stress, obviously, in this animal paradigm that induces sleep. I guess it's a certain sort of intense experience. It's not the kind of low-level stress where we're worried about whether we can get our grant application in on time and can't get to sleep, or we've had a bad or some other thing like that. So it's a very peculiarly intense, I suppose, biochemical wave that induces it following this situation. And to put that in a wider context, there are other stressors that induce sleep, such as some people call it like sickness stress. So when stress from that's got a sort of molecular explanation with interleukins and things. So I guess it's a very specific phenomenon, but it must induce some kind of intense biochemical change in the brain of these mice. And then there is, as you point out, variation in this protocol. Sometimes when people do that multiple times, the mouse gets injured. That wasn't the case in our study, and that contributes to wakefulness as well. So the whole phenomenon is, I fully admit, it's a bit of a idiosyncratic one, but I don't know, in our hands, that's what happens. Mice get stressed and go to sleep.
Yeah, that's fascinating. So from what I understand, it's not very well defined in human, right? Although I see a comment here by Vanda Clement saying that in human, stress can also induces a somnolence. But it's look like you open a window for looking at this effect in human, right? Also.
Oh yes, if it can be done on a large scale, it's one of those things that at the moment, I mean, as I said, it's sort of anecdotal. One professor, a chemist, wrote to me from Stanford, and he read our paper. He doesn't know anything about neuroscience. He just read the abstract, and he said every time there's a stress, he goes to sleep, and it's happened to his mother and one of his children, and they offered to give us a GWAS study, but it's just that their family. So intense stress makes his child go to sleep, like if they have to do exams that day or something. I mean, I've no, it's anecdotal. It's not.
Yeah. No, but I believe it's very interesting, and of course, I'm sure clinically it should be examined in detail, right? It's very important because in patients they have a lot of problem with stress, to cope with stress, and it's very nice that you see this decrease of corticosterone also induced by sleep and by the neurons you discovered in the VTA. And there is also a question, and so I had the same question in a way. So of course you induce both an increase on non-REM and on REM. So there is a question whether what is more important, the increase of non-REM or REM in the effect you report. I mean, do you have an idea about that? Of course, beside what are the pathway involved? Is there a difference for non-REM and REM?
Given the specific questions, given the specific functions to anxiety, so we think long-run sleep might contribute to the anxiolytic effect, because there are other findings showed that different stress protocol causes different REM sleep pattern. In another study, people found that long REM sleep was that has been induced after a few hours after oxidative stress, and then they did the similar experiments to measure the anxiety level, and results actually an increase of REM sleep, the anxiety-like behaviors were also reduced. So, well, at least from the perspective of anxiety, we think non-REM sleep might contribute more than REM sleep. But REM sleep is also very important for memory formation and other emotional process. So REM sleep might, after stress, might help to process other emotional behavior besides mental process of anxiety or depressive-like behavior.
Thanks a lot. I see a question from Anita Luti here in the she's online. So I don't know, Anita, if you are ready to ask your question.
Yes, sure.
Okay,
Thank you,
great to have you with
us.
Well,
thanks for the really exciting lecture and the great presentations. It has been really a pleasure. And I mean, I raised my hand right after you ended your presentation. So some of my questions have already been answered. I mean, as I think many of you, I have wondered also about, the quality of your social defeat stress compared to other protocols that also have been taken as incentives to study PTSD type behaviors. Now I see that you have even done it twice in the same mice. On the one hand, the first round to label the neurons, and then the second one to see whether you can suppress the changes in sleep. So how are these mice, what kind of baseline do you start from when you subject these mice to a second social defeat stress? Have they fully recovered in terms of their sleep-wake behavior, or are you having changes with respect to non-REM sleep partitioning of sleep?
Yes, so we tagged mice with 2 episodes of social defeat stress, and the first one is to allow tagged with the GERD receptor, and the mice had a fully recovery period for weeks. So, and then we conducted the second episode of stressor. So we think the mice, the second one actually does not, the first one does not affect the second manipulation because they have been allowed sufficient time to recover.
Very interesting.
Thanks a lot, Anita, for your question and the answer. I have a comment about these neurons. You see these neurons increase their activity during stress. The VTA GABAergic neurons or somatostatin neurons, they increase their activity during social stress, and then their activity remain high during subsequent sleep, and even the somatostatin link seems to be more specifically active during sleep. So I was thinking, is it that common to have neurons in the brain active during a stress and then during sleep, or is it different neurons? So that's my first question, because this would be quite new, right, to have these kind of neurons, but I don't know if it has been reported for other type of neurons. And my second commentary is about the efferents, the projections of these neurons responsible for inducing sleep. So I saw that you say that it's by means of the projection to the lateral hypothalamus, and I found in the supplementary multiple supplementary that you did test hypocretin neurons for involvement, and that you said that it's not these neurons who are involved in the induction of sleep. So it's not the inhibition of these neurons who is responsible for inducing sleep. So, but it's neurons who are in the lateral hypothalamus, so not hypocretin neurons. So I wonder if you have a hypothesis about the type of neurons they are inhibiting these VTA GABAergic neurons, and if you believe one type of neurons do everything, right? The activation during stress and the induction of sleep.
So the first question is about the enhanced activity of those neurons. So these actually phenomenon, I noticed that some drug, such as cocaine, can enhance dopamine or cells in the nuclear components, and with an enhanced activity for after drug administration. So one way is probably due to the plasticity stress, or those drugs remodels the plasticity of neurons, which so those cells had an enhanced activity for a few hours or even for a few days in other literatures. And another possibility is that because we found SST cells are active during non-REM sleep, it could also be sleep was induced by the first activation of those cells. And then the long-run sleep, actually, the sleep itself also triggered the activation of those cells, because in the supplementary, we showed that sleep deprivation actually suppressed the activity of the enhanced activity. And of course, there are other possibilities that sleep deprivation caused secondary response, and therefore the activity was suppressed. So, well, this remains, we think, still an open question. And to answer your second question, the effector neurons in the RH. So we tested it's not hypocretin or orexin cells, but there are other cell populations interlinked in the RH, such as VGLU2 and the ghrelin and the GABA cells. And those cell types, they have been shown their importance in sleep-wake regulation, and they could also respond to stress. So it could be any subtype of these cells, but needs to do further studies to see which cell types responsible for the VTA SST effects.
Okay, thanks a lot. So I believe we need to close the session now because we are a bit over time. So it has been a very lively session. So I see that in the question and answer there are some questions about when you will be. I don't know if you can maybe answer to them now. When you will apply, I mean, your discovery to human, because of course, yeah, Bill, maybe you want to have a concluding, I mean, remark.
Well, of course, we'd love to be able to manipulate these neurons in a way to induce sleep if it's restorative sleep. And I mean, of course, I don't have a simple solution to that, but I'd just like, I mean, Zhao discovered these cells, and we discovered them actually without a hypothesis, as Zhao showed at the beginning, by screening chemogenetically throughout the brain. So whatever they're doing, in addition to stress, this tiny population of GABA cells in the VTA, in our hands, they're really potent at controlling vigilance state. So maybe other stimuli can activate them to induce sleep as well. And so, yeah, of course, I don't have a concise, clear answer to how we can do it in humans, if at all, but maybe deep brain stimulation one day, you know, to target VTA, but, you know, of course, I don't have a clear answer, but thank you very much for the question about it.
Yeah, thanks a lot.
Sorry.
Yeah, so thanks a lot, Bill, for and Zhao to accepting to participate to this online webinar. Thank you also, Amy and Takeshi, for your presentation. So I did like this format personally. I believe, although of course it's not as lively as at physical meeting. And I hope we will see each other at physical meeting in the future. So we are setting up such meeting for Sevilla, for example. But I believe this format, and hopefully you all agree, is quite interesting to communicate and to discuss together. So it will, yeah, Bill, you have a comment, a final comment?
No, well, thank you for inviting us, and I think that's worked really rather well. Linking Japan and Europe and gone around the planet, so that's rather good. Yeah, couldn't do that without, and we haven't had jet lag, so that's great.
Yeah, you're right. So thanks a lot again. So we close the session now. Thanks a lot.
Thank you.
Thank you. I enjoyed a lot.
Thanks for listening.
Yeah, enjoy your lunch and see you hopefully soon later. Thanks. Bye bye, everybody.
Bye bye.